University of New Hampshire stretches over 2,500 acres and contains countless unique environments. From the built environments of its dormitories and academic halls to the natural environments of College Woods and Oyster River, the land is an ever-evolving mosaic of habitats. These habitats support a host of local and invasive creatures, from the thousands of students and staff that attend the school to the countless birds, rodents, and other mammals that live amongst us. Yet there is a whole other world often overlooked by our large distracted eyes, a world just as dynamic and alive as any other, the world of microorganisms. This is a world full of so many different species, interactions, and processes it could take an eternity to understand. So for now, we'll just focus on a few. A brief overview of the area we inhabit and those tiny life forms that inhabit it with us. The old bricks and stones that make up the towers and halls built to withstand and shelter against New England's many seasons may seem bleak and barren to us, but can be quite hospitable for some microbes. The roughness that gives these materials their characteristic looks provides places for microbes to attach, and places for water and nutrients to seep in. Fungi are commonly associated with the built environment, with penicillin being the most frequently found microbial genus across a range of building materials. Aspergillus is another genus of fungi commonly found in construction materials, especially ceramics and fixatives, such as bricks and mortar. Of course, stone and brick aren't the only materials used to build stuff. Metals like iron and steel are often used in structural support. These metals can be found many places around campus, but are maybe most prominent in the set of shining silver tracks that cut under Main Street up into Maine and down into Massachusetts. With railways come railway bridges, and one of these can be found just off campus in Doe Farm. Spanning a river and exposed to the elements, this bridge sits in rustling contrast to the glinting tracks it supports. Corrosion is a natural reaction that happens when metal is exposed to water, but this process can be accelerated by microbes. Bacteria that are able to oxidize manganese and iron, such as Leptothrix and Gallionella, are often associated with metal corrosion, along with those that can reduce iron, such as Shawinella and Pseudomonas. Additionally, species of fungi like Penicillium and Fusarium, who you may recognize from both their antibiotic and pathogenic abilities, can also speed up the corrosive process through the production of acidic byproducts. Interestingly, symbiotic relationships between these microbial species in the form of biofilms often have a synergistic effect, contributing to biogeochemical cascades, which increase corrosion faster than any single species could on its own. Where there is rust, there is water, and the water that flows in the many rivers, lakes, and estuaries around UNH is home to a great diversity of microbial life. With 1 teaspoon of river water sometimes holding millions of individual microbes, and community compositions varying due to many different factors, the aquatic environment can be hard to characterize. Yet there are patterns that hold true when examining microbes in aquatic ecosystems. Fine benthic organic matter, or FBOM, is a term for the particulate matter under 1 millimeter that often makes up a lot of the silt that sits suspended or settled in rivers. This FBOM is dominated by a bacteria, while fungi make up the majority of microbes on submerged wood and leaf litter. In a study of FBOM-associated microbial communities in forest streams less than 70 miles north of campus, researchers found that the diverse gram-negative phylums of acidiobacteria and proteobacteria were by far the most abundant microbes. When taking into account more studies, stream water is generally dominated by the gram-positive actinobacteria phylum known for its filamentous morphology, along with the gram-negative phylums of proteobacteria and Bacteroides, which are often rod or pill-shaped. Interestingly, pH was found to have a strong relationship with the variability within these river microbial communities, and was the best predictor of FBOM-associated community composition when compared with other biotic and abiotic factors. Of course, these other factors are important too, and things like temperature, dissolved oxygen content, and river flow rate can have big effects on microbial composition and dynamics. As a northern state, the changing of the seasons affects all of these factors. As temperatures drop, dissolved oxygen can increase, while flow rates decrease and eventually stop altogether as ice creeps its crystalline way across rivers and lakes. While these winter landscapes cloaked in snow and ice may look desolate to us, many microbes are well adapted to survive under these conditions. In fact, while ice coverage has been shown to decrease bacterial abundance, It has also been shown to increase overall microbial diversity in freshwater environments like the Great Lakes. This increase in diversity might sound surprising, but there are a couple of things that could explain it. It's possible that the selective pressure of the winter freeze could inhibit certain taxa that dominate in warmer water, and thus provide opportunities for more diverse communities to take over. Similarly, the ice cover might kill off predators and provide a respite from microbial grazing. Or this winter diversity could be due to an increased presence of dormant cells that become more detectable as dominant summer taxa decrease. This is something that can happen when using methods that show relative abundance of taxa, such as 16S amplicon sequencing. While dormancy is certainly a strategy some microbes use to survive freezing, many have been observed to continue active growth throughout these conditions. These include algae like cyanobacteria, which have been shown to progress in slow-forming blooms during periods of solid ice cover. Furthermore, microbes involved in nitrification can be especially active during these times of winter freeze, driving important seasonal biogeochemical cycles. While all this life continues on under the ice, winter blusters on above, driving animals indoors or underground and rendering the landscapes in grays and whites. Yet even something as seemingly pure as snow can support microbial life. Condensation, whether it be rain, hail, or snow, always begins its journey as water molecules aggregating around a condensation nucleus. This nucleus can be inorganic dust, or, as recent research has uncovered, it can be microbes. This is known as bioprecipitation, and it seems that some microbes may use this as a way to disperse themselves across terrestrial environments, hitching a ride within falling raindrops or snowflakes. These microbes, sometimes called ice nucleators, can be fungal spores or bacteria. One common ice nucleator is a bacterial species called Pseudomonas syringae, whose unique surface structure can mimic ice crystals, and thus act as an effective condensation nucleus in snowflakes. Once the snow has reached the ground, it can act as an insulator for the soil microbial communities that live below, many of which have been shown to tolerate frozen soils. These communities are able to continue microbial activities such as respiration, which is an exothermic process, meaning it releases energy in the form of heat. The heat released by this is potentially enough to warm surrounding soils, which in turn can increase microbial respiration and thus contribute to a positive feedback loop. These seasonal freezing periods are also important in things like nutrient leaching, mineralization, and decomposition, as well as supplying dead cells that can support microbial life and lead to an activity spike as things begin to thaw. Spring brings many changes and can be a time of plenty for macrobes and microbes alike. New substrates are exposed, organic matter accumulates, and frozen water is released into its more easily accessible liquid form. Like any good forest, College Woods has a lot of trees. These trees are dependent on soil microbes for much of their growth and survival. Many of the leaves dropped in the fall re-emerge along with hibernators as the snow recedes. Microbes are essential in breaking down these leaves, freeing important compounds stored within them and replenishing the soil. With a single gram of soil often containing billions of microbes, there's a huge diversity of species involved in these processes. Bacteria, including many members of the Proteobacteria phylum, are often associated with breaking down nitrogenous organic matter, while fungi, like Ascomycetes, are often associated with breaking down carbon-based matter. Underneath all this decomposing litter is a vast expanse of wandering roots in their microbial symbionts. Mycorrhizal fungi play a big role in the decomposition process by releasing enzymes and exudates that support the soil community, but they also play crucial roles in tree growth and interactions. They can do this by forming mutualistic relationships with the roots of trees and other forest plants. This is an incredibly widespread relationship that virtually all trees are thought to participate in. Ectomycorrhizal fungi form a kind of sheath around these fine root networks, which can benefit the trees in many ways, including providing protection against various environmental stressors, helping in nutrient breakdown, storage, and uptake, and perhaps most famously providing a potential signaling and transport network between individual trees, although the specifics of this are still being researched. As a mutualistic relationship, the mycorrhizal fungi also benefit through access to a steady supply of metabolites produced by their tree symbionts. These underground networks stretch far and wide in support of the many woods and forests about campus, but are far from the only habitats around. Located only a dozen or so miles inland from the Atlantic, the surrounding waterways and wetlands are not all fresh water. Great Bay Estuary and the many tributaries and salt marshes between us and the coast offer even more unique microbial environments. On a broad scale, the estuary is dominated by many of the bacterial phylums already mentioned in freshwater environments, including Proteobacteria, Bacteroidetes, and Actinobacteria, as well as members of Chloroflexi and Firmicutes. These mix with marine microbes in an environment that selects for plankton able to tolerate the salt content and other chemical gradients and tidal variability associated with coastal waterways. When environments are mixed like this, it provides an opportunity for native communities to develop. For this to happen, the microbial doubling time must fit within the time that microbes remain in the area, which is important to take into account with the tidal dynamics of an estuary. This is known as residence time and can change a lot depending on specific geography and season. In the Great Bay Estuary, the average residence time has been measured between 5 and 20 days, suggesting plenty of time for local communities to develop. Local microbial communities support local macrobial communities. Everything from mollusks and crabs to ducks and herons have associated microbiomes. In these symbioses, microbes can benefit from a constant source of nutrition and protection, while their microbial hosts benefit from increased digestive capability and immune function. Additionally, while motile appendages like flagella and processes involving physical and chemical gradients like chemotaxis may allow microbes to move over relatively short distances, longer travel often requires outside help. This can come from things like wind, rain, and tides, or it can come from relationships with larger, more motile animals like fish, humans, and birds. In fact, interest has been growing in recent years over migratory birds' involvement in spreading microbes, especially those that can cause disease in humans. Antibiotic-resistant human pathogens like strains of the gram-negative bacterium E. coli have been found in the guts of many coastal gulls known for long-distance migrations. Some suggest that these strains are picked up while gulls feed at sites contaminated with human waste, such as landfills and treatment plants, and are later dispersed along coastal regions in the gulls' droppings. The sheer amount of microbes surrounding us, all the relationships they're involved in, and the processes they affect can be hard to fathom. From the vast underground fungal networks regulating the health of our forests, to the ever-shifting algae colonies driving nutrient cycling in our seas, to the airborne bacterial constellations influencing everything from our health to our weather, microbes really are almost everywhere. And while our eyes might not be able to see them, our bodies and environment are constantly feeling their effects. Some of these, like disease and decay, can be quite painful, yet these same effects in other contexts can also be incredibly helpful. In the end, we can associate microbes with much of the good and the bad that make up our lives, and know that without them, life as we know it would not exist.